Okay, we may have a few more join us here in a moment, but I want to introduce to the group Amy Kamara.
Hi.
Amy is Assistant Professor Cropping Systems at Vernon.
Extension agronomist.
Extension agronomist at Vernon. Okay. Well, I was fairly
Nice to meet
close.
you.
I had your name
Yeah.
and the location correct. Anyway, welcome. This is a plant breeding brown bag seminar series we call the Plant Breeding Circle. If you would like to join it by TTVN or whatever our electronic stuff here is called, we can make that arrangement. You can join us from Vernon if you would like. Just let me know,
Okay.
and we can work on that and make it happen.
Okay.
But,
I'm just sorry.
Yes.
Our population is really nervous because the biggest variation I've seen is in maturity.
Oh,
Unless you're
I'm not surprised by that. Because what I'm worried about, you see that variation.
All right, we're going to go ahead and get started. I just have a couple of announcements to make real fast. We are currently full for the rest of the semester for Plant Breeder Circle. I am going to start scheduling next semester here in a little bit, so if you would like to present next semester, come find me, or I will start finding grad students to present also. I think Brian has an announcement to make in terms of...
This is for graduate students interested in plant breeding. Laura Macer started a new plant breeding organization with students, and they had their first meeting yesterday. So the second meeting ever is going to be on October 29th at 5:30 here on the 4th floor of Heath. So we encourage anybody that's interested to come to that. And if you're on Facebook, you can get updates about the club by joining the Facebook page. Mitch.
What's the club do?
So it's brand new, so that's the great thing about it is we're not really sure what we're going to do yet. We have three main objectives. One is to build a sense of community between graduate students studying plant science, plant breeding related things all across the different departments in the university. We also plan to do professional development type stuff, and also advocate for the needs of graduate students in this area.
Is this the Plant Breeding Graduate Student Association?
Yes.
Okay.
All right, thank you.
Yeah. All right, and then for today's speaker, we have Drutdaman Bhangu. He's going to be talking on the impact of natural drought on extra long staple upland and fiber strength traits in South Texas. And he is one of my cohorts in the Cotton Institute lab.
Thank you, Lauren. As Lauren said, the topic of my presentation today is impact of natural drought on extra long staple upland and fiber strength traits in cotton in South Texas. So there are two cultivated species of cotton in the United States. First one is Gossypium barbadense, second one Gossypium hirsutum. Gossypium barbadense is usually known as Pima cotton, is also referred to as extra long staple cotton, precisely because of its really high upper half mean length, which exceeds 35 millimeters. The other species, Gossypium hirsutum, is medium staple upland, which accounts for about 95% of cotton production in the U.S., and its upper half mean length ranges from 27 to 29 millimeters. So why improve fiber quality? Well, the first reason is we will have improved product if there's improved fiber quality, and with improved fiber traits, strength, uniformity, and other quality measurements that go along with fiber quality. And the second one is to remain competitive with man-made fibers. And man-made fibers or synthetic fibers are increasingly getting better at fiber quality. The way we're going to approach to improve fiber quality is obviously through improved breeding methods and techniques. One being the genomic techniques such as using markers, SNPs, and genome-wide selection. And the other one being standard Mendelian techniques. So the material that we're going to use, or that we did use, was Texas A&M Con Improvement Lab material. One of them was extra long staple Upland, where the upper half mean length exceeded 34.9 millimeters, which is about 1.37 inches. Long staple Upland, where the upper half mean length ranges from 32 to 34.9 millimeters. And then another one, which is extra strength Upland, which fiber bundle strength exceeds 36 grams per tex. And this was all the three strains were developed without the Gossypium barbadense introgression. A little bit of background on extra long staple Upland. As I said earlier, they were developed without the barbadense introgression. TAM94L25 was the progenitor of the ELSU trait and a common parent among the 5 families of extra long staple Upland. The source of this improved fiber bundle strength is not really well defined. The possible explanation is that it resulted from an incremental improvement using pedigree breeding, recurrent selection breeding, and multiple parents since about 1970. So the objective was to compare the stability of extra long staple Upland, long staple Upland, and strength traits in Texas A&M genetic backgrounds along with current cultivars when grown under irrigated and natural drought in South Texas during 2013 and 2014. The genotypes, the first one is extra long staple Upland with having high fiber length, that is TAM11K13. The second one, extra long staple Upland with the high strength strain, TAM11T08, and the third one being long staple Upland, TAM11L24. And they are from the elite quality cotton breeding program at A&M. The other TAM strains come from Dr. Steve Haig's program, TAM07B45, TAM08WZ78, and TAM08WZ83. And the remaining 10 entries were commercial cultivars that were entered in the variety trials in 2013 and 2014. The experimental design, it was a randomized complete block design with 4 replications. The 2 locations that we used for our study was Corpus Christi and Weslaco. Corpus Christi was dryland, and Weslaco was irrigated, and normal cotton production practices were followed, including boll weevil eradication. And for the fiber quality data, we hand harvested 30 open bolls and ginned on a 10-saw lab gin. The lint samples were evaluated at the Fiber and the Biopolymer Research Institute at Lubbock at Texas Tech. And HVI fiber properties were determined, which includes upper half mean length, micronair, strength, uniformity index, and elongation. The statistical analysis, lint percent was determined as lint weight divided by seed cotton weight, multiplied by 100, and then lint yield per hectare was determined from machine harvested plots. And GLM procedure of SAS was used to determine the differences in fiber properties and lint yield with year, location, and genotypes being fixed effects. We also used the USDA loan value chart for 2013 and 14 to determine the value of lint per kilogram and the value of lint per hectare. And they were adjusted according to any premium or discount using 3 categories: micronair, upper half mean length, and strength. And also in the loan value chart, which is divided according to the color grade and the leaf grade. And we had color grade of 41 and leaf grade of 4. So in our ANOVA results, as expected, year and location, there was significant interaction because we are using 2 different locations and 2 different treatments, as in irrigated and dry land. But What we are really interested in was the genotype by location, where we saw a significant interaction in upper half mean length, strength, and uniformity index. And we used upper half mean length and strength as our basis for to look at the stability of it in both the locations. Uniformity index is, we didn't look at it because it doesn't go into the lone value chart. There is no such system that looks at uniformity index and determines your lone value of the lint. To look at the stability of the three elite strains when compared with commercial cultivars, we looked at the delta percentage values. Now these values are derived by subtracting your dry land value from your irrigated and then divided by the irrigated value so that we can see what percent change was there when it went from an irrigated situation to a dry land. And you'll notice that there is a significant overlap among all the genotypes. And in the elite strains, there is a significant difference when they go from irrigated to dry land. There's a 19% change, and then the long staple upland shows a 16% change. However, that is not a significant, that is a lot less numerical overlap between all the genotypes that are listed there. So we can see that there is stability among the elite strains when compared with commercial varieties when they are grown in irrigated and dry land situations. Similar pattern was noticed in strength. There is a bigger numerical difference between the strength traits. But if you look at the elite strains, they had the least amount of percent change when they went from irrigated situation to a dry land situation, and they only showed about 2.3%, 0.7, and the last one, the extra strength upland, TAMU 11 T08, actually increased on average its strength when it went from irrigated to dry land. So in dry land, it performed better on average when it comes to strength trait. So this table tells us that there is a lot of stability with the elite varieties when compared with commercial varieties, especially in dry land situation. In this in this table, we look at the lint value per hectare based on premiums and discounts that are in the USDA loan chart. In the first quadrant, we notice the loan value, that the elite strains had a really high loan value. And that year there was a severe drought, so the lint yield, you can compare it with the other quadrants, that it's significantly low. And it performed really well in 2013 in dry land situation in Corpus Christi. So it was quite competitive when compared to the commercial cultivars. But if you look at irrigated in 2013, the elite strains again had a really high loan value. So did the commercial cultivars. However, it took a dip in the yield. It performed considerably, it had a considerable lower yield when compared to the commercial cultivars. And a similar pattern was observed in 2014 in dryland situation as well in Corpus Christi. Once again, the loan value is the highest, 1.25 for the elite strains. However, the value, the dollar value for those elite strains is really low compared to the commercial cultivars. And same with 2014 with the irrigated in West Coast. Less yield, and that's why there was a less $1 per hectare value for the elite strains, even though they had a good loan value. So in conclusion, the extra long staple upland and the strength traits exhibited stability across the environments compared with commercial cultivars. The strength trait was maybe more stable under moisture stress than expressed by current cultivars, and the two extra long staple were the only genotypes that averaged above the upper half mean length, above the minimum for PIMA when grown under irrigated culture. TAM 11K13 and 11L24 exhibited significantly smaller decreases in fiber strength when grown under drought stress. There was a small percent change, as we saw in the table. In TAM 11T08, the extra long staple upland produced numerically stronger fiber. Its fiber strength increased when it went from irrigated to drylands location, a phenomena that we have previously observed, but we don't know what's causing that. And the last, despite a higher loan value in stress environments, yield potential and lint percentage still remain as challenges in developing extra long staple upland cultivars. Now I'm going to talk about my research a little bit, so I can just a couple of slides. Even I don't have any. I do have a little bit of data, but I'm in the process of gathering all the data and all the analysis, and then I'll have in spring to present on my master's thesis. But I'm going to present a little bit about my work and hopefully get suggestions from all of you to further improve on my materials and methods or techniques that I'm using. So what I'm working on is chromosome substitution approach towards introgressing novel traits from Gossypium barbadense into Gossypium hirsutum. Gossypium barbadense is the Pima cotton with the really high fiber length, with the really high quality, and trying to introgress those traits into hirsutum upland cotton, which is normally the highest grown cotton in acreage in the United States. So chromosome substitution, where one chromosome of the Gossypium hirsutum recurrent parent is replaced with the chromosome of Gossypium barbadense donor parent. Dr. Stelli's lab have developed those 17 chromosome substitution lines, also called disomic alien chromosome substitution, the CSB lines. And the hirsutum parent that is used is TM1, Texas marker 1. It's usually a reference genotype that is used, and its upper half mean length is 27 to 29 millimeters. And 379 is the Gossypium barbadense parent, where upper half mean length is higher than 35 millimeters. So the basic research question that we're trying to address or answer with my research is that are major fiber quality genes that are expressed phenotypically in barbadense commercial cultivars, are they located on a specific chromosome? Because chromosome substitution lines can help us identify which particular chromosome is adding to the increase in fiber length. And also, could chromosome substitution lines aid in establishing SNP or other molecular markers for fiber quality? So the materials and methods. First part is the diallele analysis. I'll use a line by tester design to screen for chromosome substitution lines that produce the F1s above the average of all the chromosome substitution lines for length, strength, or yield potential. There are 17 chromosome substitution lines that I'm using, with the two parents being TM1 and 379, and three testers. TAM P18233 is extra long staple upland, being used for its fiber length, high fiber length, and TAM 06WE 621 being used for its really high strength, and TAMCOT 73 being used because of its really high yield. And the location is College Station. The F1s right now are being grown in 4 replications. And then the F2 population will be grown to evaluate ranges and variances selected for selected fiber traits and the initiation of recombinant inbred lines for future genomic analysis. And the fiber data analysis will include high volume instrument and APIS, the Advanced Fiber Information System. The second aspect of my research will include QTL detection, the high resolution mapping of F2 progeny to detect QTLs. And so the F2 progeny, so far what we have decided the mapping that we're going to do is going to result from a cross between from the 2 of the testers, TAM B182 being the high fiber length one, and TAM 06 being the strength trait. And we're going to select 30 to 50 plants. We're going to extract DNA from the seed of the 2 crosses that will be selected for mapping. And then markers will be selected based on the SNP chip, and then the SNP markers hopefully can provide for QTL detection in the chromosome of Pisidium barbadense. This is sort of the rough sort of map that what I'm going to do. And with that, I would like to acknowledge my committee members, Dr. Wayne Smith, Dr. Steve Hagan, Dr. Stelly, and Con Improvement Lab, Don Dino, Richard Hermes, and all the grad students and the undergrad students who helped me conduct this research, and also Dr. Stelly's lab, and Dr. Fei Wong for the DNA analysis, and all the graduate students at the Dr. Stelly's lab too. With that, I'm open to any questions or suggestions that you guys have.
So is there a market for longer fiber?
Is there a what?
A market for this longer fiber?
Yes. Well, in most of the cotton produced in the United States is exported, and the rest of the countries or the rest of the world look for a high fiber length, and they look for about 35 millimeters as being 35 is the staple length, and the fiber length in America is around 34. So higher fiber length is an advantage for the farmers, not only because it will give more money, but also in export it can drive more production, or it can.
Yeah, the only reason I asked is just because I keep hearing things like it's the yield that matters and not the.
Sure, yield does matter, but I think we should constantly be improving the fiber length so that we can compete with the international market or with the man-made fibers or the synthetic fibers. Yes.
I was just wondering, how many markers are within the chip you're going to be using, the SNP chip? Do you know what the number of markers
I
are?
don't know. I think Dr. Stelly can answer that question.
There's 19,000 between
Okay.
Pisidium and barbadense.
So how confident are you that you're going to, with 30 to 50 individuals, doesn't seem like you're going to have much power in detecting QTL?
Well, that's the constraint that with the time that we have, and also with the money as well.
Okay.
I'm sure you can, if you want to do it, you can,
Yeah.
You can widen your.
Just gonna write a grant between now and then and have $14 million. We're gonna expand it.
So it's more constricted, just based on field space and funds for the number of lines you're going to.
Yeah, and also I think it costs a lot to use the SNP chip
So I think you mentioned doing seed DNA. Is that what you said? Did you say you were going to use seed-derived DNA?
Yes, from the last discussion I had with you.
Yeah, so you can use that for doing CASP assays, not chip. So the quality is not high enough for doing chip analysis. You don't need much DNA for the chip, but you need to have high quality.
The quality of DNA would be not that.
You'd be better off using more conventional type DNA prep. which is more time and more work.
Go ahead, Mitch.
You can intercross or shoot them in Barbaden's, correct? Isn't that the same thing as chromosome substitution? Why are you using chromosome substitution?
Well, then you can, let's say you had an increase in one of the F1s with the, with the fiber length that had a significant increase, then you can at least go back to that particular substitution line and hope that it's that substitution that's causing that increase in the fiber length. You narrow down your, I guess, the.
So what's the rationale in sticking, you said you're going to only use the F1s that were higher? Did you say that or not?
Yes.
Why?
Well, we're going to look, in the line by tester, we're going to look at the F1s. Well, the obvious, because we're comparing them to substitution lines, I think if any of the F1s perform higher than substitution line, then perhaps it's, if it's a good-looking F1, then hopefully that'll be a good-looking F2 as well.
Yeah, but you're constraining yourself to codominant, dominant beneficial effects. So what if you have recessive gene combinations, for example, that come from your two parents? Is that?
Sure, I don't.
Might want to think about that one. And then I guess another question would be, I know we talked about doing chip analysis, but if you're only looking for segregation within one chromosome, it might make more sense just to buy primers and run those SNPs for one chromosome. So most of the chromosomes are 100 to 150 centimorgans in length. So if you pick out 10 or 15 of those, you get pretty good coverage. And you're just looking, at this point you're not, I don't know if I would call high resolution at that point, but you're going to pick up on whether or not you have a major effect or not. And if you see something, you can always come back with additional markers in that region. I don't know, it might be worth thinking twice about how you want to approach the markers, and then also making pretty good progress on GBS, and that's interspecific. So that might be another route to go. I'd keep my options open a little bit on that. But there too, if you're going to go to GBS, you need high quality DNA. If you're going to do the Cas markers, yeah, that's PCR based. You can get away with the cDNA extraction. That'd be fine.
Or we can grow out the F2s in the greenhouse, hopefully, if the cDNA is not the right option.
Yeah. So, could I ask one question about your table 2 and your first?
Yeah.
I was curious, so W is Westlaco,
Yes.
and CC is Corpus Christi, and
Yeah.
then you have your delta values, which are the differences between the two locations.
Yeah. Well, we set they're arranged according to the absolute delta values, so which had the highest change in the absolute value.
But the changes between locations, right?
Yes.
So what you're really interested in, I would think, would be differences between your dry land and your irrigated within a location, isn't it?
Depends on what you want to predict it over, right?
The locations are the dry land and the irrigated.
Oh, you just had one in each location?
Yes.
Okay, I got you. All right. For some
Yeah.
reason I was thinking,
Yeah, Westlaco
you
was
had one of each in each location.
No.
Yeah. Westlaco was completely irrigated, and the dry land was entirely Corpus Christi.
But that makes sense.
So we talk for the students, we talk a lot about interactions, and the GBI is important, and you'll find paper after paper that to identify the G by E, they list the means at one location, they list the means at the other location, and they try to compare them, how they change in rank. And certainly you can get away with that. But if you're looking at the interaction, it is how a genotype behaved in the two environments, the two either in this case locations, which we said were, one was dry land, was irrigated, both in South Texas, so sunshine's about the same, et cetera, et cetera. There's a lot of other variables, but the major variable was moisture. And so you can take the difference between them, and the mean separation would have been on the value, not on the percentages. But the only way you can statistically separate those interactions is his column called delta, or the difference between the two. Then you can apply an LSD using the error mean square to calculate that, changing it up, the formula up by, now it's 4 means instead of 2 means, and you can separate that column that he has called delta. For the interaction, there's no way to separate the stuff from an interaction perspective under the two columns of locations or two environments. And so if you're looking to identify, and to a breeder, that's the major thing you're interested in is my variety stable across some number of locations or some different set of environments. And it will get pretty complicated if you have 12 environments, 10 locations, and 10 more treatments or whatever else. But from a separation of interactions, I think that's the only way you can separate them. Someone can challenge me on that one.
My varieties were all stably low yielding, so I moved into administration.
Stability is not always a good thing.
Yes, Carl.
So I've got a question to, I guess, continue on this table, and it's for you or either Dr. Smith. But in the high strength material, that phenomenon that you said you mentioned of higher strength and I guess unfavorable or rain-fed conditions, is that a consistent effect that we see across the different high strength germplasm lines, or is it specific to a few? And do you have any kind of hypothesis on
what's causing that?
What kind of physiological mechanisms are going on?
I would say that maybe without moisture, the fibers are a little, they have a high micronair, so it's a little too coarse. I don't know. That's what I would think. That it probably has a lot more cellulose. Probably the drought or lack of moisture is causing more cellulose in the secondary cell wall, and there's a lot more. Perhaps that's what causes the fiber bundle strength to go really high.
But just in those genotypes?
I haven't looked at any other genotypes that have sort of this sort of stability or this sort of interaction in dry land or in irrigated.
The short answer is we don't know. I have seen it in some other situations. Never tried to quantify it. This is the first time we had put any kind of come close to quantifying it. But it does. I've seen it enough to think that it's not unusual when you get strengths up into the 36, 38 grams per text. The other teachable moment for you other students is it's okay to say I don't know the answer to a question.
Not all the time.
Not all the time. When you don't know, you say I don't know. Do you know? So.
I need in your first study,
yes.
First presentation, you mentioned that barbidancy has 35 kind of mm length, and hirsutum is 25 to 27.
27 to 29.
Something like that. But the stem lines are hirsutum and have 35 mm length.
They get to that
Yeah, you have some lines now that have 35 mm lengths, and if barbidense has 35. And on Wednesday, there was a presentation that fiber length is highly heritable trait with very little G/E interaction. So what you have in your first study, why do the second study? Why go to barbidense if you already have that highly heritable trait? Why not breed with that and run with it, or just put the high yielding gene? Why
Well,
worry about second study?
Well, we're interested in the behavior of the trait, and if pinpointing fiber length, let's say that fiber length is maybe coming from a particular chromosome is adding to the increase in fiber length.
No, my question is why not stick to hirsutum itself when you have the same range? I go to barbidense. Just unless you are interested in chromosome manipulation, that's a different objective. But if your objective is the fiber length, you already have within hirsutum, run with it, do whatever you want. Why complicate your life? No, I mean, this is something that...
Because I want 1.5 and 1.6 instead of 1.3, 1.4.
Yes, Dr. Hayes.
Damon, so based on this information, you were designing a breeding program. How many different environments would you select for fiber? 1, 2, 3, every location?
Well, if I have the money, why not include a lot of locations?
No, the most value, most bang for your buck.
From a science perspective.
If you're setting up a commercial cotton,
Yeah.
how many different environments would you select for? How many different environments would you use to select fiber?
Two.
Which two?
One being dryland, one being irrigated. And maybe what Dr. Selly was suggesting, maybe have multiple locations but have dryland and irrigated right next to each other, if that's possible, just using all the locations for dryland and irrigated instead of just using one being irrigated and one being dryland.
I will say this, my opinion would always be rankings are not really going to change that much, but based on this data, my thoughts are a little bit different now. But I also wonder, because some of those that didn't change very much in strength, they also probably don't put that many bowls on the plant either. So the source sink may not be. I don't mean to be bashing your line.
Yeah, you're going to start talking about me. Just get no respect. Rodney Dangerfield, the breeding rules.
But I am curious if that would play into it.
I have one more question on this second study. You have 4 environments, 2 locations, irrigated and non-irrigated, yeah? So I will consider that as 4 environment.
But he only has 2.
I don't
No.
2 years and 2 locations.
2 years, 2 locations.
Yeah, but irrigated and non-irrigated.
No.
No. One being irrigated and the other one being.
Oh, so
Yes.
that was confounded with location.
It is confounded.
That's what threw me off.
Yeah.
Yeah, that's...
So how many genes are in cotton?
I don't know.
Gee.
How many are expressed in cotton fiber development?
465? That's what Dr. Hong Bin was
That's
talking about.
how many he says are important, and then he can
In
account
length.
for absolutely 100% of the variation in the trade. But there's something in excess of 13,000 expressed in fiber development. Of course, those 465 or whatever it is could be anywhere in the plant. They don't just have to be in the fiber, right? Unless that's all he was analyzing. But plant performance has to affect fiber development as well.
Yeah.
So anyway, so let's say there's 465. That's quite a few genes, right?
Yeah.
Why would you expect to find, talk about your second study, why would you expect to find significant differences tracing back to individual chromosomes from another species? If you change one of those genes, let's say, that's 1/400th, that's less than 1%.
Sure, you can see negative I mean, you can see fiber length decreasing as well. Perhaps the chromosome that was taken out maybe was adding to the increase in fiber length. You know, the hope is because we have barbadense having higher fiber length, that's what will add to fiber length. But it can possibly, maybe the genes in that chromosome that was taken out maybe were responsible for over adding to higher fiber length.
So I'm being difficult here for a reason. So if you change one chromosome, how many genes are you changing? If you swap one out from hirsute and replace it with one from barbadense, how many genes are you supposed to change?
I don't know. Unless we have recognized, unless we have found out every single.
We have a pretty good idea. I'll let you read about it, figure it out.
So what if you ask that another way? VB suggested, or someone suggested, that you just simply cross hirsutum and barbadense, and you're using Dr. Staley's chromosome substitution lines. So how many alien genes are you not having to deal with if you use a chromosome substitution line relative to just crossing with barbadense?
But you're certainly reducing with using substitution lines, you're reducing all the other extra material that is coming along if you're just using hirsutum and barbadense.
By how much? You say you're reducing how much?
I don't know. We'll have to look at.
Oh, you know.
Yeah, you can figure it out. How many chromosomes are there in cotton?
26.
Right? And you're swapping out one of them, so how many of them are basically identical?
Or hirsutums?
But
25 out
25.
of 26.
25 out of 26.
Over 95% in theory. There's some residual background. These are PC5 derived lines, but it's a large percentage. So somebody was asking about, well, why not just use an F2, regular F2? And that's really the question that Dr. Smith is alluding to. You have a tremendous amount of genetic noise that you're trying to discern what the effects are of specific loci within one chromosome when you have tons of stuff segregating all over the place. Now there's some advantage in having that stuff, so it's kind of good to do both, but in terms of just seeing effects of individual loci per se from that alien chromosome, that's a great advantage to have everything else pretty much isogenic so that you're just seeing the effects of that one chromosome. So you're having a chromosome-specific F2, right? It's not segregating basically for other loci. There's some, but by and large, you're segregating just for the genes out of that one chromosome, which means you can discern what their effect is much better. And so going beyond this a little bit, we're also developing chromosome-specific RILs, which has been done in wheat, for example, previously. And that's even more powerful because then you can run replicated experiments on those derivatives. And so you can go after traits that are not just the highly heritable traits. You can go after things that you need replicated experimentation for.
Can you go back to the slide with your experimental design for the mapping in the F2, the chromosome substitution line?
Yeah.
So you're going to look to map both length and strength, and that's why you're using two populations?
Yeah.
Because you have such a low number of F2s that you're screening within those, so your power is going to be reduced.
Yeah.
Have you given any thought to, say, just focusing on maybe length or strength,
yeah.
and then expanding the number of progeny you look at with population so you have more power?
Sure, that was considered in the discussion. Actually, we actually had thought of all the three. The third one being the yield one, time code 73, and then just we thought that maybe we'll get more out of doing two instead of just focusing on one. But then that's also a good point that in order to increase more of your, I guess, efficiency and mapping, just looking at one of the interaction in just one of the F1s.
So how much power will you have if you say 50 plants? How much power do you kind of feel that is? In terms of mapping out a quantitative look. How much recombination would you expect to have?
I don't know.
So if each chromosome is, let's say, 100, 150 centimorgans, how many crossovers do you have per chromosome per plant, per F2? So if it's 100 centimorgans, that's at least 2 crossovers per chromosome. You got 2 chromosomes per F2, so that's at least 4 crossovers per plant. So you've got 200 crossovers, and you've got each chromosome is going to average 100 to 150 centimorgans. So you have more than 1 crossover per centimorgan. It's pretty powerful. Even so, and the power here is in part because of, again, of this isogenic background. You're not dealing with all the other garbage, or as much of it. It's a reduced garbage situation. It's not perfect, but it is powerful. And you can see this if you go to the wheat work that's been done. So Todd Campbell, for example, did some of this stuff in wheat when he was at Nebraska with Steve Benson here long ago. So it's a very powerful approach, and it's going to be interesting to see how this plays out. I would probably analyze both traits for both crosses, because it's going to, on one case, each parent you're bringing in lots of, or some, additional positive genetic variation for a trait you're interested in, and the other one you're not, right? And so there's been some top cross work done with chromosome substitutions before. It's done with a group at USDA in Mississippi. And there are variety by variety interactions that you see with the specific chromosome substitution lines. So you're going to see differences between these, I would think. It's quite likely.
And we now have lines that have both those traits in the same, that contain both those traits. If we were starting over, we'd have only one combination instead of 2.
So a big question is, how much of the barbadense variation is already in here soon, right?
Yeah, and there's some historical speculation that there's
right.
that the 2 species are already commingled alleles.
Right.
So, but they still, someone asked if they were interfertile, and yes, they are, but they tend to segregate back out to type as you go over the generations. Right. No one's ever been able to stabilize an intermediate between hirsutum and barbadense, particularly for fiber properties. You cannot. No one's ever crossed barbadense or Pima with Upland and selected an Upland phenotype that had barbadense fiber on it.
Yes, it goes along with the idea you have lots of genes, and some of them are going to be in big pericentromeric regions that don't have any crossovers, so you get one or the other, that's it.
Yes, okay.
Your economic analysis, when you put the market loan value to the cut, what year did you use?
2013 and 14.
Okay. It would be interesting if you went back and you did today's market loan value, because it's about 60 cents.
Oh.
Because what you could do then, because there are a lot of extension people running around saying how important it is, fiber quality is when prices are low, you would be able to put a number to that and demonstrate, is it really that important when prices are low? Or are we still chasing pounds primarily?
When we calculated, it was 52 cents a pound. And you're saying that it's 60 cents?
So you use 52 cents a pound?
Yeah, we use 52
okay.
cents.
Okay.
It's already at the low end.
Okay.
We hope.
All right, because I thought I saw values of over $1 per pound.
Yeah. There were, because we used all the,
Yes, $1 per kilogram.
Oh, okay. You were using metric.
Yeah.
Okay. So we're using the euro in there as well?
It's time to break.
In one of your slides, I think materials and method, you showed that you will use dialyl analysis, but actually you had line by tester.
Yeah.
Are it both or dialyl? I mean, it cannot be both.
That's right.
Is it line by tester or dialyl?
Yes.
It's line by tester.
So forget about dialyl then.
Okay.
What he meant to convey was you'd be looking for combining ability.
Yeah, that's
using
fine.
line by tester.
All right, if there are no other questions, let's thank Damon for
One little announcement before I turn it back over to him, and that is that got word yesterday that we have a research farm at Commerce again. The land sale went through yesterday, and so Dr. Dr. Harp will be down in the next couple of weeks to visit with us about how we might plan to utilize the new land there. Okay?
All right, you guys, a lot of your colleagues didn't show up today. You need to get on their case.
Yeah, it's getting clear now. I would